So regarding the future of this, what is next? I mean, you just outlined really, quite honestly, fascinating points of this. Where do we go from here, and what are the advancements you're talking about? If you could refer to each of the three trials you're talking about, that would be great.
Yeah, absolutely. So, where do we go from here? I mean, I think, so far the trials to date, all three of them have shown that CRISPR gene editing can be used in a safe fashion. So remember, that's one of the concerns that emerged when CRISPR was first discovered, the ethical implications of editing a genome. Can we use this in a safe manner? And I think, although it's a limited data set, I think we have enough data from these three different trials to suggest that CRISPR is, in fact, safe if it's used in the appropriate context. The second thing to highlight here is I think there's going to be tremendous application across all three indications or all three disease areas that I mentioned. So, for example, in the context of monogenic disorders, which is these are inherited disorders caused by mutation in a single gene. We've already seen quite a few trials emerge where they propose to use CRISPR gene editing to overcome diseases like Tay-Sachs or congenital blindness. So these are folks that are born with a mutation that causes them to be blind. So there's proposals to use CRISPR gene editing in that context. In the context of cancer immunotherapy, people are coming up with more creative approaches for trying to use CRISPR gene editing to enhance the cancer immunotherapy. So there's a trial that's recently opened up. It's being sponsored by the National Cancer Institute and by the University of Minnesota, where they propose to knock out a checkpoint gene called CISH. And what they hope to do is enhance the potency, the efficacy, the activity of the cancer immunotherapy to be able to overcome that. And so that's, only made possible through something like CRISPR. And then in the context of infectious diseases, we're going to see more trials like the HIV trial. But I think another application is going to be using CRISPR to enable the development of diagnostics. So, a traditional diagnostic, like, if you want to see if you're positive for COVID or HIV or something else, it requires pretty elaborate equipment, like a PCR machine. And so a lot of times places like, less developed countries don't have access to electricity or some of these sophisticated devices. So what people have developed using CRISPR gene editing is diagnostic approaches to enable you to figure out whether you're infected with something, but something as simple as a at-home pregnancy test. So it's like a color detecting reagent, and that's only made possible through something like CRISPR. So the point is that I think we're going to see more of these types of clinical trials, but also we're going to see CRISPR expand into other areas of healthcare beyond therapeutics, like diagnostics and other areas as well.